Thank you everyone for taking the time to attend our webinar today. Hopefully you'll find it interesting and please, as Bob said, ask questions. We'll be very pleased to answer them. So I'm going to introduce the topic, the background and our speakers. I'm very pleased to have speakers from both REPROCELL and also from FiosWebinar Genomics. Just to briefly introduce myself, I'm the CEO at REPROCELL Europe, based in the UK. My background in pharmacology and around 20 years in the CRO industry. Firstly, co-founding Biopta, which was acquired by REPROCELL in 2015, doing contract research work for pharma and biotech companies in the area of human tissue testing. And my colleague Graham, who is Director of Precision Medicine, formerly head of R&D, also at REPROCELL. Graham has more than 15 years experience in the pharma biotech industry, designing, delivering preclinical projects. And since 2011, when he joined REPROCELL, has been leading a team developing a range of tissue-based assays, in particular around areas of inflammation such as IBD, psoriasis and COPD, which we'll touch on today. Also pleased to be joined by Dr. Max Balesio. Max has been a collaborator of ours for a while now and has been a pleasure to work with him in his role at FiosWebinar Genomics, where he's technical director. He joined FiosWebinar in 2012 and is now on the board of directors. A bioinformatician by training, Max is responsible for the operations group. In addition, he oversees R&D of new analysis methods and IT infrastructure. He has a PhD in chemistry and MSc in engineering biology from Sweden, and also previous commercial experience with Systemic and Almac Diagnostics. So today we're going to have Graham and Max are both going to explain a very interesting project that we've done together. And REPROCELL, you may not know, is a company based in Japan, but with operations in the US, Europe and India. And it's one of the leading stem cell and human tissue focused product and service companies. We're going to talk mainly about the human tissue research that we do through the labs in Maryland and in the UK. But we do also have the facilities focusing on stem cell research and genomics in Japan and India. I'm going to explain one very important area for this talk, which is around the use of human fresh tissue, because it gives some background to a unique approach that we've employed for close to 20 years now through Biopta and now through REPROCELL, where we obtain ethically sourced human tissues from surgery or transplant, and we use those as models for testing new pharmaceuticals. And this workflow shows a schematic of how we actually do that. We obtain the tissue, could be heart, lung, or in the case of some examples today, it will be COPD, lung, but also tissues such as skin, inflammatory bowel disease, psoriasis, bladder, many types of human tissue that we can access fresh and transport to our labs in Maryland or in the UK. What we then do is use those fresh samples as living tissue specimens to test the effects of new drugs. So these are some of the closest models that we can possibly study to understand the potential safety and efficacy characteristics of new therapeutics. This is typically used as powerful translational data to understand how cell-based assays or animal models translate into humans by giving us that early stage preclinical data in actual tissues from the target patient population. Here we can see an example of a drug where we're looking at the effects on blood pressure by investigating the responses of a small blood vessel, and that would translate to a potential effect on blood pressure. That concept can be applied to almost any tissue type and endpoint. And Graham will explain the approach that we've taken for this project that we've done together with FIOS. Now, one of the main developments for us since we first launched this service back in 2002 is that precision medicine, personalized medicine has gone from being a concept to becoming an integral part of the strategy for pharma and biotech companies. Part of that is because we're all much more aware now that many compounds that are already on the market are not effective for a large percentage of patients. These are a couple of studies from the UK Technology Strategy Board, which is a UK government institute back in 2010, and then more recently in Nature in 2015, showing the percentage of patients that respond and don't respond to common medications. Now that aspect of trying to improve the selection of a medicine for a patient has very much been focused on genomics and understanding genetic differences between individuals. But it's also now widened to understanding pharmacology and relating pharmacology in individuals to genomics. And that's really going to be the subject of our presentation today. So Graham's going to explain how we actually use the pharmacology and how we can relate that to genetics. And this has actually been done previously in a couple of studies that we were involved in, which I'm quickly going to describe, just to give you an example of the power of this type of analysis. One of the tissues that we look at in the lab are isolated bronchi. So we're looking at airways in the lung and measuring constriction or dilation in response to test drugs. So these could be new drugs, or then we may use standard of care drugs to give a comparison. So here we've got two concentration response curves to standard of care drugs here, positive control drugs, formoterol and salmeterol, both acting at the same receptor. And we've studied that in 3 patients. So the same 3 patients in both graphs. And you can see for formoterol, very similar responses across 3 patients. But for salmeterol, we've got quite different responses. We've got effectively a good responder, a responder, and a poor responder. And in fact, that measurement that we see in the lab using a fresh human tissue is reflected in the clinic. That's what's known to occur and is a major problem for physicians when they are trying to treat asthma, in particular childhood asthma, where often children will be given salmeterol, and then a few months later, parents will come back and say, it's not helped, the child's still wheezing and still having symptoms. And this is now known to be related to a specific mutation in the beta-adrenin receptor that affects the responsiveness and sensitivity to salmeterol, but not to other beta agonists. So we can relate the pharmacology using these fresh living tissues to the genetics potentially, and that's something that we set out to explore further. And part of that is because pharmacogenomics has become much more important to the development of new drugs. The cost of genomic studies has fallen, and also there is a missing link in understanding human pharmacology at an earlier stage in drug development process. So I'm now going to pass over to Graham, who's going to explain how we've tackled this in the current project.
Okay, thanks, David, and hi everyone. Okay, so I'm just going to take control of the screen for 1 second. Okay, so following on from the background that David has just kind of talked us through, I am kind of firstly going to really summarize firstly why REPROCELL believe that a human tissue can help to fill that a need and gap, and then also how it can further these precision medicine strategies that are taken at a much earlier stage than would kind of standardly be done. The kind of problem which is outlined at the top of this slide is obviously well known, as David said. It is kind of well understood now that not every drug responds in the same way in every patient, and one of the solutions in our perspective to this is to use human tissue assays at an early stage in kind of drug development to do that. These human tissue assays are highly complex, and whilst they have their limitations, as all systems do, the complexity of them is highly important here, as it really allows them to be representative of the local disease biology. Using human tissue-based systems, such as primary cells or tissues, whilst it allows the efficacy to be estimated, which has been shown in kind of some of David's slides, it also allows the combination of that data to be looked at and analysed alongside the associated human data, which is shown on the right-hand side of this slide. Things like the kind of donor, a demographics and medical history that's associated with the a patient sample that's been received, and also things like the genomics and transcriptomics that are also associated with that individual tissue. And it's kind of really this combination of data and the advanced analysis of it that adds value, and that also gives it the potential to really allow us to better inform patient selection for kind of clinical trials moving forward, and really to allow us to do it at a much earlier stage than in a current kind of common practice of trying to stratify the patient population post an all-comers a style of kind of clinical trial. Okay, so what types of associations can we kind of look to draw from this type of data and these types of projects? And on this slide, I've tried to highlight some kind of potential associations that can be made. From up at the top left-hand corner here, if we combine the donor medical history along with the kind of pharmacology data, we can start to look at, for example, how do patients who have been on a specific drug, such as steroids, respond to a novel drug? Okay, or in the bottom left-hand corner, if we combine the patient medical history and the pharmacology to then look to see how patients from a kind of specific cohort of a disease respond to drug X, or even if we look at the a medical history itself, so how does a patient with, say, kind of COPD or IBD, what kind of SNPs are associated with that specific patient cohort? There's kind of various different associations that can be made, and these are really just an example of some of them. Okay, so that kind of really summarises our concept and the theory behind it, but how does that actually work in practice? And to help demonstrate this, I will now kind of move on to describe a project that we conducted in collaboration with FIOS and some other companies, academia, and also the NHS in Scotland to help exemplify the approach that we've just described. Okay, so what were the main objectives of that collaboration? Well, okay, firstly, we've already described that overarching goal was to try to demonstrate the ability to better improve that patient stratification strategy, prior to clinical trials, and using human tissue assays and the associated omics and patient data to help do that. And then secondly as well is really to aim to showcase the strengths of the precision medicine capabilities that are present in Scotland. Scotland really offers the ability to combine world class life science companies, academia, and also the NHS and kind of research projects such as this, and also with our life science companies, the kind of commercial experience and expertise to perform these. I'm going to keep this a presentation quite top level about the project, but if anyone is interested in learning a bit more detail about it, then we have also published on this work, and it can be found in the kind of Cowan et al. Paper, which is on the right-hand side of this slide. Okay, and it's an open access article which can be found in PLOS One. It was published in December 2019. Okay, so about the project itself, how was it? How was it actually kind of structured, and how did we work through the project itself? Firstly, obviously, it has to begin with a patient. So a patient, as David said, who is perhaps having kind of surgery for a procedure, and and is going to have a piece of tissue removed, and kind of some of that can be used for research, then when a patient can consent for that to be used. Once we receive the tissue, there is a basic donor information that comes as standard with these types of tissue. And then like that tissue and information can be used in various different ways. Like firstly, like a human functional tissue pharmacology experiments like those that David explained in the slides earlier. And that was performed at REPROCELL in the labs in Glasgow. And then also with the collaboration with NHS Scotland and via their data safe havens, which includes all the electronic health records of people in Scotland, then we can also have access to detailed donor demographic information above and beyond the norm. A portion of each a piece of tissue that we received was also used to extract some RNA. So here for this project, we extracted RNA and using a microRNA specialist company, kind of Systemic, that partnered with us. They performed some microRNA arrays to look at the microRNA expression. Exactly the same concept could have been done with, say, RNA-seq element to look at kind of transcriptomic expression overall. And then also NGS. And so for the a project that I'll talk about today, it was whole exome sequencing. And this was performed by the Precision Medicine Scotland Innovation Centre, which also helped to fund a lot of this work as well. And they work in conjunction with kind of Thermo Fisher, reagents and instruments. And then also a company called Iridia, who has a kind of secure data platform, helped to really to manage and to store safely and securely all of the resultant data that was generated kind of throughout these different types of analyses. And then finally, then there's the important fact, which is pulling all of the data together, and really that's where kind of FIOS helped to do that, and kind of Max will describe that in more detail later. And then also for this project as well, we had the support of Professor Colin Palmer from the University of Dundee, who offered his expertise on the kind of pharmacogenomics around especially kind of lung disease, and that helped to kind of aid the bioinformatics analysis. And really the outputs of this system is a patient stratification strategy to some extent. Okay, it is a limited in data numbers, but we do get some early a proof of efficacy in a complex human system. We are able to look towards the identification of genomic markers of drug efficacy. We can do the same for transcriptomic or microRNA markers, and then also use the donor demographics as well to see if there's any associations within the data sets. Okay, so for this project, we focused on a chronic obstructive pulmonary disease, which is also known as COPD. And therefore, to measure the drug efficacy, the human tissue model that we used in this case was a REPROCELL's human lung explant assay, which uses lung tissue obtained from patients who have been diagnosed with a COPD. So these are disease patient samples. And really, the selection of the test system is kind of critically important in the kind of validity of any further and downstream output. Okay, so any patient stratification outputs that are generated, really hinge on this, because really the human test system acts as a proxy for patient drug response and disease biology. We believe that the kind of human explant system in this case was the best system the one to use, as it's using primary tissue. It has the complexity of lung tissue, it has the cellularity, the different cell types involved in lung tissue. And really the aim of the assay itself was just to maintain that function for a long enough time to allow us to add some drug treatments in there, and then to measure the modification of the inflammatory processes within that piece of tissue. There's multiple endpoints possible. Here in this project, we use the cytokine measurement, so cytokines, chemokines that were released into the surrounding media. But we could also potentially use another endpoint, okay, such as one of the a gene expression endpoint, okay, for an example. One thing to note about these systems in general, as in all in vitro systems, these sometimes require a kind of stimulant in order to maintain the disease relevant drive within the model. And in this project, we used the stimulant LPS, which is lipopolysaccharides. And the reasons for doing so, I'll explain on the next slide. Okay, so whilst we focused on a COPD, we also chose to focus in particular on examining the effects of drug treatments on treating the inflammation associated with bacterial exacerbations of COPD. And this is actually the most common type of COPD exacerbation. And whilst a treatment of the bacterial infection is obviously of kind of critical importance, the associated inflammation is also a significant cause of mortality. LPS itself is a bacterial endotoxin, and via a TLR4 agonism acts as a pro-inflammatory stimulant. So it's therefore, highly relevant to the disease biology. Okay, it's a key factor in kind of choosing your model system and any added stimulant. And then another key and important choice in any model of efficacy is that what measurement or measurements you're going to use in order to determine drug effectiveness. And for this project, we chose to look at the pro-inflammatory cytokine like TNF alpha. This cytokine has been reported to be measured in kind of sputum samples from patients who are suffering from both COPD and also elevated following exacerbation, and has also been kind of shown to be a really good and predictive a biomarker as such of airway bacterial infection. Okay, so that's why we chose to measure a TNF here. The a graph on the left-hand side of this screen actually kind of shows some of the data we generated kind of pre-study to really to validate the use of LPS in the system and to ensure that it was a suitable stimulant. We took 10 donor samples, okay, and we kind of split the resultant lung biopsies into two groups. One group that was media only, okay, so no stimulant added, and then there was a group that had LPS added, okay, and then we cultured them out over a period of time, and then measured the amount and quantity of a TNF alpha that was released from these biopsies, and that's what's measured on the y-axis. Each of the matched donor measurements here are linked by a line. So as you can see, there is, although there is already quite a high baseline level of inflammatory cytokine TNF in this case, as the sample was diseased, adding in LPS also further increases the levels of TNF, which is in line with the observations, from sputum samples that have been taken from patients. Okay, so really that was the main a reasoning behind choosing these as drivers. And then also for TNF itself and LPS, it's also important to take into account the mechanisms of the drugs that you want to test in the system. It's very, very important to make sure that any expected mechanism used is also in line with the way that you're either kind of stimulating culture or measuring within your system itself. Okay, so now moving on to the actual pharmacology data that we actually generated. Okay, there are various options here, and how you design this part of the experiment, it's really up to what you want to get out at the other side and what you wish to study. You could, for example, look at just one drug alone, look at the effect of a drug over a range of concentrations in order to get an understanding of potency differences between patients. Or, as we have in this study, we're aiming to look at really the difference in maximal efficacy, okay, across a range of drugs and or combinations of drugs. And in these experiments, we used 25 donor samples, okay, so kind of 25 individual COPD donor lungs were used, and we tested the following drugs. So firstly, we tested kind of roflumilast, which is a PDE4 inhibitor, an anti-inflammatory compound, at a concentration of 100 nanomolar. And this is a standard of care treatment for COPD in adults, especially with adults who have frequent exacerbations of their COPD. When we also tested the corticosteroid fluticasone at a concentration of 1 micromolar. And again, kind of fluticasone is a standard of care treatment for COPD and is often used as a maintenance therapy to suppress levels of inflammation. There has, however, been reported a kind of wide range of either initial donor insensitivity to steroids, or over time an increased or a kind of decrease in sensitivity to kind of steroid treatment. So that's why we chose that one in this study. And then the third, like a test compound, was kind of formoterol. Okay, so a beta agonist. We tested that here at 10 nanomolar. And again, it's used in COPD, like regularly for patients. It's not primarily used as an anti-inflammatory, like it's used to kind of relax airways. However, there has been reports in the kind of literature and hypothesis around that may have some anti-inflammatory effects in certain patients. And it's also been reported that it can add to or kind of increase the anti-inflammatory effect when dosed in combination with some anti-inflammatory drugs. And in particular, there are some literature around PDE4 inhibitors. So again, that's kind of why we included it in this study. Both graphs on screen are both actually representations of exactly the same data. On the left, we have the mean data. So this is the mean of the results of the 25 donors, plus or minus the standard error. And as you can see, we are getting an expected reduction in the level of TNF. So for each of these drug treatments, they're compared against the DMSO control. All of the drugs in the in vitro system were made up in DMSO. So that's why we're comparing back to a DMSO control here. And that's the 100% value. And then each patient is kind of measured back against their own a DMSO control condition. So with the anti-inflammatory compounds, we're seeing a nice decrease in the levels of TNF. For kind of formoterol, we didn't really see an overall mean decrease, although there were some patients, when we look at the scatter plots on the right, that maybe did show some anti-inflammatory effects on their own. And then again, we saw a nice decrease with the combination of roflumilast and formoterol. Okay, so that's the mean data on the left, but what that actually hides is the variance in that data as such. So when we plot that as a scatter plot, which is on the right-hand side here, each individual dot here is the mean result for an individual donor. Okay, so each dot on here is one donor, and that's why there's 25 dots for each test condition. And as you can see, there's a kind of variance across the test conditions and also within test conditions of how well each of the donor samples has responded to each a drug treatment. And that's something that Max will discuss in a little bit more detail on how we kind of dealt with that type of data from a bioinformatics standpoint. Okay, so that really kind of summarizes the overall concept of the study we conducted with a particular focus on the human tissue pharmacology. I'll now hand over to Max at FIOS, who will describe the bioinformatics strategies employed to analyze the data and also how it was associated with other human data streams. Okay, Max.
Great, thanks, Graham. Hi, everyone. I'll start from that to just with a quick introduction about FIOS Genomics, which is the company that I represent. And we've been involved in the bioinformatics support for this interesting project. So FIOS is a Edinburgh University spin-out, which was formed back in 2008. We're based in Edinburgh's BioQuarter, the building on the right. What we do is bioinformatics services. We don't have wet lab facilities. We're kind of pure service provider in the bioinformatics space. We work with a range of data types and providers, mostly with pharma, biotech, both CROs and academia. The area that we work in is mostly drug development, preclinical and clinical, with a range of omics data types, which is something that we'll discuss as part of this presentation. So high-dimensional data like transcriptomics and genomics data. Our focus is very much on the biology and understanding the biology behind the data, and not just on crunching numbers as such. Okay, so just to add to what Graham already introduced in terms of the assays to generate the TNF-alpha readouts across the different drugs. On top of having those for 25 patient samples, there was also profiling done with targeted exome sequencing for genotyping, microRNA expression profiling just to measure the full microRNAome, and then at the end, it's just to integrate that with the drug response data that we have to understand what are the drivers and what can explain the effects that we're seeing really between the different types of responders. So the plot you see here is a similar one to what Graham showed earlier, just with the TNF-alpha readouts, and these are normalized against control, so the control is always 100%. And by this definition, I think all of these samples are pretty much responders. They fall below that line, so they have suppression of TNF-alpha release effectively. But that by itself does not mean that all of them have the same type of response. As you see, that there are certainly differences between them. So the question then becomes, how can we identify and use this information for the purposes of patient stratification? And how can we link that back to the other types of data that we have? The first port of call is to look at differences between the efficacy outputs for TNF-alpha, and specifically looking for bimodality, which is when you have two different distributions or two different peaks. So the plot here that's shown on the left is the output from the treatment of roflumilast and fluticasone. So we just picked that one and we looked through all different outcomes of drug treatments here. This is the one just for the case study, just to illustrate what it looks like as the one that has the most distinct profile that we're interested in. So in this case, you can see that what looks like two slightly separate distributions, which can be categorized as two different types of responders. So all of them in effect are responders, but some of them are slightly stronger. So this plot is flipped compared to the one I showed earlier, and the high responders also have the strongest suppression TNF-alpha on the left, and colored in red, and then the remaining ones are the low responders on the right, which have a slightly lower suppression of TNF-alpha in that group. So once we got to that point, I think we have two potentially different groups of responders that have a different levels of TNF-alpha readouts on average. The first question perhaps to ask, once you have that, is there something that we already know that can easily explain those differences? So for instance, are the high responders of a different type of age or gender or ethnicity? And that information is provided as part of this study, as Graham illustrated earlier. So we can look back over that and see if there's an association. So what we've done is a statistical test effectively to see whether there are associations between different pairs of factors. So for instance, is the high-low response criteria associated with age and gender and ethnicity and so on. In fact, we did across the board in the plot on the right, just showing the pairwise associations between different factors in this study. And some of them are coming up, for instance, age and ethnicity are associated. But at the bottom is the bar for the response to roflumilast and fluticasone treatment, which is not associated with anything in the study. So what that means is that we can't explain this separation based on the clinical and demographical factors that we have available, which leads us to the next question: is what can actually explain this, and are other avenues to understand why there is a separation in that drug outcome. So working through the data types that we have, microRNA data set is the first one where we have a full microRNA expression levels across all these patients. So can we utilize that information from patients to explain the response differences, and how do we actually do that in practice? If you're not familiar with microRNAs, they're small non-coding RNA molecules. They function primarily for RNA silencing for mRNA. In this case, we have 24 patients profiled using the adjuvant human microRNA microarrays. The workflow that we're looking at for microRNA data analysis is in a general sense, just quality control and exploratory analysis. So that's just to understand the quality of the data, look for potential QC failures, look for structures and patterns in the data sets, and see if that matches expectation. And then there's the statistical analysis for differential expressions. In this case, very much a targeted question of what's different between the high and low response groups in this case. And then we take the differentially expressed microRNAs and try to piece them together into pathways. So to look if there are any particular pathways that are coming up stronger than you would expect by chance, and then try to understand the biology around that pathway. Is it a relevant pathway? Is it something that you can link back to the original mechanism of the treatment? And if that's the case, can you take that forward from a further biological understanding? To sum up the microRNA results, then the quality control and exploratory analysis, we performed normalization and batch effect correction in this case for profiling batches. A high-level summary is that out of the 24 samples that were profiled, 23 passed quality controls. One sample had an aberrant profile and we decided to exclude it from downstream analysis. If you look at the plot displayed here on the right, which is a principal component representation of all the microRNAs in the set, each point here is one sample, and they're colored by the response criteria, so high and low, the definitions that we already outlined. Samples are close to each other here are expected to have a similar microRNA profile. You can see there's no strong stratification due to response group. That by itself does not mean that there's no signal in the data to describe differences between them, but at least the main driver in the expression set is not directly linked to response. So to start off, then we focused our questions a little bit more and asked whether we can find differential expressed microRNAs in this set that are different between the high and low responders as defined earlier. The data set has only 25 samples, and in the context of big omics studies, that means that we have a relatively low power to detect differential expression. So we were a little bit cautious when it comes to interpretation, but taking into account the patient numbers, we used an uncorrected p-value of 0.05 here. And uncorrected in this case means it's not correcting for multiple correction and inflation coming from that. And at that level, we had about 181 microRNAs that were significant at this threshold. And those are the ones on the right in the volcano plot, which are highlighted in red and blue. And they're roughly evenly spread out between those that are up and down regulated, in this case, between the high and low. So from those 108 microRNAs, we then did pathway analysis to understand what the microRNAs were doing. So microRNAs are frequently annotated through their target genes. So rather than the microRNA itself, you look at the potential targets, or as in this case, from the confirmed as experimentally validated targets. We use the MERTAR base to look up that information. And then you take those genes and look at the significant enrichment in certain pathways. And so we looked across a set of pathway databases to perform that type of analysis. And one of the main outcomes from this was that there was an enrichment in the TGF-beta signaling pathway. And when you look into the detail of why that's coming up, it's mainly coming from a SMAD3 regulation, which is a target of one of the significant microRNAs in here, which is MER18A. And if you start looking through the literature a little bit and try to figure out what's going on in that system, it's clear that TGF-beta and SMAD have been shown to be implicated in the pathology of COPD. It's also clear that it's something that's known from roflumilast treatment in particular, that has a regulation in this pathway too. So based on that, it was an interesting finding, and I think from follow-up studies, we also managed to back that up with additional support of previous findings to explain the context and the relevance of that effect from the study. So that's the first part of the omics jigsaw. The other one is genotyping data. So we also have genotyping data, so can we use that information to explain response differences? And the samples here from the full 25 samples with short read sequencing using the Ion Torrent platform, which is based on whole exome sequencing. The workflow for genotyping data is largely quality control, alignment, and variant calling. So that's kind of the heavy lifting when it comes to the pre-processing. And then we look at statistical analysis to identify associations with response groups. So very similar to the microRNA in the sense that we're asking the questions, what genotypes could explain the differences between the high and low response groups. Just a quick summary of the upstream processing. So I think all the samples here are of good quality, passed QC. We ran them through the standard Torrent alignment, and the reads mapped to a sufficient level, which you expect based on the number of reads we started with. We have variant calling with Torrent variant caller. and I ended up with around 37,000 variants for each sample on average. And most of these are single nucleotide polymorphisms, and so it's just a single base pair that's being affected. So we took that information, so we have the defined variants, so the mutations in these samples as compared to the reference, and then we try to associate that, so to find a way to explain how that links to roflumilast and fluticasone. You can do that with a statistical approach, in this case called Fisher's exact test. That will give you an indication of how strong the association is between the genotype and the phenotype, which is in this case the readouts from the response groups. We also know, again, from the sample numbers that we have to be a little bit cautious with interpretation of results, and lacking the full power of major studies. So we looked at an uncorrected p-value of 0.001, and this is purely to have some results to look at in the context of pathways and processes, and also link back to the microRNA. And with those settings, we have a total of 30 SNPs that came back significantly mapped in a unique set of 23 genes, and they were significantly associated with the drug response. And a number of these, in fact, have been reported to be associated with COPD or other pulmonary diseases. A couple of them I'll just outline in the next few slides. Well, you probably see that SMAD3 is one of them, which has already been highlighted through the microRNA study. So that adds another level of support of the relevance of that particular gene and pathway. So here's SMAD3 as one variant within that gene that was identified with a level of significance as outlined before. So the genotyping information is here illustrated in two different ways. So you have a mosaic plot on the left, which outlines the genotypes versus the response criteria. So the way to interpret that one is to look at the genotype, which is listed at the top. So you have the reference, which is AA here, and if you follow that column down, you'll see that all of it is blue, which means that all of the AA are low responders. And the same for AG in this case, whereas the GD column has a mix. It's primarily high responders. You can also see the same information tabulated on the right, which just shows how the number of samples are spread across them. So in this case, you can say that all the low responders, or the AA and AG genotypes, are always low responders, whereas the GD is more spread between the two, but predominantly coming from the high responders category. And so that's like an interesting finding. It's not necessarily conclusive in terms of explaining everything, but it will give you a good indication of association. Another way of displaying a similar thing is to look at the original TNF-alpha readouts. So rather than just bundling them together as high and low responders as we've done before, you can look at the same values that we had to define the two groups and overlay that with genotypes. In this case, the lower values here indicate a stronger response. And you can see the same information, really, that the GD genotype in particular has a much lower TNF-alpha values, and that's significant in one comparison here. The remaining one lacks a little bit of sample numbers to reach significance. Another genotype came out quite strongly and also had a link to the mechanism of COPD as one of the cytochrome E450 genes and proteins. So this is CYP2E1. Similar representation here on the left, the mosaic plot just shows you the reference here, which is TT, has all high responders, so that bar is completely red. And it's a bit of a mix between the other two, but predominantly high responders in the heterozygous TC and predominantly low responders in the homozygous alternative allele, CC in this case. And it's tabulated again on the right, is showing how that's broken down based on the different genotypes and the response criteria. And that's again not a fully separating the two response groups by genotype, but it's certainly a strong indication and a significant association in this case. Similarly, you can also take the original TNF-alpha values and look at those. In this case, you see quite a nice trend going from low numbers, which is the TT reference, which is a strong response, going up to CC, which is the alternative allele, where you have a much higher value of TNF-alpha. And that kind of gradient is quite interesting from the perspective of understanding what's going on and linking genotype to phenotype. So to sum up those results, look at those the small pilots here, which are 25 Genomics vivo COPD samples, and looked at the readouts from TNF-alpha to understand how patients respond differently to different drugs. And we can see that individual patient responses were quite different in some cases, and you can also match them against genotype microRNA. And based on that, you can find some interesting associations, which in the case of 2 genes in particular were backed up by previous findings in the literature, and also had suggested links to the drug treatment or the disease mechanism in most cases. And now further information is available in the publication, as Graham mentioned earlier. And that's the end of the presentation. Thanks very much for